Hi, my name is Kris Engelstad. I am a clinical researcher and genetic counselor at Columbia University in New York City. I became involved with Glut1 in 2001 when I interviewed for a job and received it with Dr. DeVivo in his clinical research center. At that time, he had already seen patients with Glut1, and we had a natural history study for patients with Glut1. A natural history study is a study whereby we follow people over time. We don't give them a study medication, we don't treat them with anything, we don't interact with them in any way to change the disease course. So we really just wanted to know what would happen over time in patients with Glut1. Now you have to remember that Dr. DeVivo discovered the disorder prior to that time, and he'd seen a few patients. But there really wasn't a lot known about Glut1 at that time. We knew a few basic things, we knew some of the phenotypes, but we hadn't seen a wide variety of patients. And many of the people that we saw were people who were very severely off. So people that had severe movement disorders, they had cognitive deficit, they had, again, difficulty walking, difficulty speaking, and just really overall general sort of significantly disabled. So at that time, it was very important for us to find out what would happen to them over time. And actually, throughout the many years that we were involved in the research, we are seeing more and more patients who have very mild Glut1. And this is very different than many of the patients that we saw originally. So it's been very useful to meet with all these patients, and we've seen well over 100 patients with Glut1. And I know that we continue, and there's many researchers in the field today that continue to find different types of patients with Glut1. So it's been very interesting to see all these patients throughout the years. So I'm involved in clinical research with Glut1. I'm also involved in clinical research with mitochondrial disorders, which I'm not going to go into today, but those are some of the things that I do in my daily work. And we have, again, natural history studies in the two disorders. We've had clinical trials, and a clinical trial really is some type of intervention with the patient. So in our case, it's been with study drugs. So that is a clinical trial. You bring a patient in, do all the outcome measures, see how they're doing, their movement disorders, give them a treatment, and then follow them over time to see, are they getting better, are they getting worse? So those are some of the things that I've been involved with in research, along with contact registries and other various types of surveys, really kind of a wide variety of things. I am also a genetic counselor, as I mentioned, and I serve that role for a pediatric neuromuscular clinic in neurology. And much of what we see are patients with pretty sort of common disorders. There's a number of neurological conditions that are more common. You obviously wouldn't probably see them in your general day-to-day, but being in neurology in a clinic, we do see a number of those disorders. So I serve as a genetic counselor for that clinic. So let's talk a little bit about some of the basics for genetics for Glut1 deficiency syndrome so that we're all on the same page, we all understand the same thing. So basically, Glut1 deficiency syndrome is caused by a genetic mutation in the SLC2A1 gene. This gene is very important because what it does is it brings glucose into the brain. So the reason you feel like you have energy in your body is because you're eating food, you eat carbohydrates, you eat fats, you eat proteins, but the most important thing for creating energy, not only in your body, but also in your brain, are carbohydrates. So carbs would be things like pasta, your ice cream, some of that you're eating, sugars, fruits, and it's important that that energy, which is in the form of glucose, gets into your brain to give your brain energy. And the job of the SLC2A1 gene is to make Glut1 protein. And the job of the Glut1 protein is to bring that glucose over the blood-brain barrier, because it's not so simple to get into the brain for any chemical. And it'll bring the glucose into your brain so your brain has energy. Now, the reason you would have Glut1 deficiency syndrome, or you might have, is because you have a genetic mutation in the SLC2A1 gene. Genetic mutations in most of the patients that we know of are de novo, meaning that the patient themselves is the only one that has the mutation. There's nobody else in the family that had Glut1. And there is, like I said, most of them are de novo. Each individual has 2 copies of the SLC2A1 gene, one that they receive from their mother and one they receive from their father through the egg and the sperm. So if just one of those copies has a genetic mutation, then you will, and the mutation is harmful, then you will have Glut1 deficiency syndrome. So one thing we want to remember about the genetics of Glut1, and remember that I said that in Glut1 you just need one mutation in one of the copies of the SLC2A1 gene to have Glut1 deficiency syndrome. And really the body requires 2 functioning SLC2A1 genes to make enough Glut1 protein to be able to bring the glucose into the brain. So if you don't have enough Glut1 protein being made, then you more likely will have Glut1 deficiency syndrome. So this is a condition known as autosomal. It's an autosomal dominant pattern, which means, again, that only one mutation in one of the copies is enough to cause the disorder. Now, those mutations can be transmitted to the offspring. So if someone with Glut1 wants to have kids, they need to be clear that there is a 50% chance for each child of receiving or inheriting the mutation from the mother or father, whoever happens to have Glut1. So 50% chance, or 1 in 2, for each child. The percentage doesn't change if you have more kids. It's the same thing for every child that you have. So that is called an autosomal dominant inheritance pattern. Now, we've also seen, but it's very rare, in patients, we've seen a few patients that have autosomal recessive patterns of Glut1 deficiency syndrome. And in that case, both of the parents had carried a mutation in the gene. Both of them were fine because the mutation was very mild. They were still making enough Glut1 protein to be able to function properly and have all the energy they need. But what happened is that the children inherited both copies of those mutations. So mild mutations in the parents are both carriers, and in that situation, each and every child has a 1 in 4 chance of receiving both copies of the mutation. Likewise, each of those children has a 1 in 2 or 50% chance of being a carrier like their parents, and also each child has a 1 in 4 or 25% chance of not inheriting either one of those mutations. So again, this is very rare. I really only know of 1 or 2 cases where we've seen this, but it can happen. And just remember, if you are an individual with an autosomal recessive Glut1, that each time you have a child, you will pass a mutation down to each child. So it's important to test their partners in the future. So in genetics, I mentioned about de novo mutations and the fact that most often in Glut1, the individual that has the Glut1 deficiency syndrome is the only one that has it, and we never find the mutation in mom or dad. But remember, when we test mom or dad to see if they carry the mutation, and again, if we ever tested them, we would test them for the mutation that the proband, a proband is the person who has the disorder. We would test them, it's called targeted testing. And we would test them for that mutation. And most often, neither mom or dad carry it. Once in a while, we see a very healthy mom or dad and they're a carrier, but it's very, very rare. I think I only know of a couple of cases. But what we're not testing for when we test mom or dad, because mom or dad are concerned about having obviously another child, and is that child going to have Glut1? But we're not testing the germ line. Now the germ line is the egg and the sperm that mom or dad have. So it's possible that we can have germline mosaicism, and what that means is although in the blood of mom or dad They don't have the mutation, but somewhere in their egg or their sperm, the mutation developed. And then when they had a child, that child was the one that received that mutation. So those are things that we really don't test for, and we don't have good figures as far as what percentage of parents might have germline mosaicism. And I'll talk a little bit later about testing egg and sperm, but for right now, we really don't have very good figures as far as the percentage of people that have germline mosaicism. One other issue we think of in families, and we do know of a couple of families that have more than one child with Glut1. Usually those are parents who have Glut1, or somebody with Glut1 had a child and passed the mutation down. And we sort of make a general estimate, even though it's perfectly an estimate, that that individual, when they receive or they inherit the mutation from mom or dad who's ever affected, will be about of the same phenotype. Phenotype is just your clinical symptoms, your severity, how you sort of react or relate as far as cognition and physical features. So we sort of make a general estimate that the individual that inherits the mutation from mom or dad will be about the same as far as clinical severity. Now, obviously, there's lots of things that go into any gene and how a gene functions. There are gene-gene interactions. There's things that upregulate and probably downregulate the Glut1 gene. So we can't be absolutely certain. And in fact, the few cases that I know of where a parent with Glut1 has had a child, there are little differences. They're not usually huge, but there again, I would never make the statement to somebody that if you have a child with Glut1, that your child is going to be exactly the same as you. We would make some rough estimates, but we wouldn't say that anybody would be exactly the same as in their clinical phenotype as their parents. So, some questions that people ask is, are there any unique features to Glut1 as far as genetics? So there's not anything special about the Glut1 gene and the genetics of the Glut1 gene that we need to be really concerned about. There are, in the human genome, there are types of genes that have different types of mutations, but those don't really apply to the Glut1 gene. I call it the Glut1 gene because that's kind of the slang term, the local term. The actual technical term is SLC2A1, but I'm using them in both manners. It's the same thing I'm talking about. So if we think about some of the tests that we get back and some of the genetic reports that we receive from the genetic companies when we send out a blood sample or to see if somebody has a mutation in the Glut1 gene, the most common type of mutation that we receive, they're called Missense Mutations. M-I-S-S-E-N-S-E, Missense. And what a missense mutation is, I'm going to back up a little bit, I'll tell you a little bit more about genetics. But if we think about what a gene is, and gene is G-E-N-E, it's not our jeans that we wear, but it's G-E-N-E. Genes are made out of DNA, and DNA really is the coding system or the recipe whereby everything in our body is made. All the information that we need to make every protein and everything in our body is in our DNA. And the DNA is composed of individual little nucleotides. And the nucleotides come in 4 types, A, T, G, and C. Just, believe it or not, 4 types of nucleotides form everything about us. But those nucleotides are arranged like the letters in a book. So if you think of a book, there's all these letters and they form the words. And regarding our DNA, every 3 nucleotides codes for 1 amino acid. The next 3, like the letters in a book, code for the next amino acid, and so on. There may be thousands of nucleotides, there may be hundreds of amino acids, but all those amino acids are hooked together like the boxcars on a train, and that's what makes up the protein. And again, the protein we're talking about is the Glut1 protein. So when we have a missense mutation, what happens is that just one, believe it or not, just one of those little nucleotides switches, just usually by chance. The human body is amazingly complicated, and when cells grow and divide, things can go awry, and if something goes awry in a gene that's really important, like the Glut1, then you can have significant symptoms. So let's say our first amino acid the nucleotide should read ATT. Again, those are our 4 nucleotides, ATGC, but our first one is composed of 3 nucleotides. And let's say that instead of ATT, it read ATC. So by accident, a C was placed whereas T should be. And because of that, or our other nucleotide changes, that single amino acid that should have been placed into, like I said, the boxcars on the train, should have been placed into the length of the amino acid change. Then what happens is that amino acid is something different, and either the protein is not made, or it's not functioning right, or it may not be functioning at all. It may actually read, the message that may be read is that stop making this protein. And then you get no protein at all from that one of the copies of the SLC2A genes, SLC2A1 genes. Remember, there's a second one, there's a backup. So one little tiny change in a nucleotide can cause Glut1 deficiency syndrome. Now, one issue with this is when we send the blood off to get a genetic report, these little missense mutations, sometimes the lab absolutely knows that it's a mutation and they report it as such. Whereas other times what happens is that they know there's a change there, they know there's some type of mutation, but they don't really know if it's causing anything. Is it really harmful? Is it really pathogenic? They don't really know. So the report would come back called a variant of uncertain significance. And I know many people have gotten reports such as these back. And in that case, sometimes it's important to follow up with some other way of trying to determine whether or not you have Glut1 deficiency syndrome. And sometimes the neurologist or the geneticist will say, well, we found this mutation, I'm going to say that this is what's causing the disorder. And sometimes what they want is additional testing, maybe a lumbar puncture to see if there's low CSF glucose. And as you remember, I mean, I was around many years ago when the only thing we had was the lumbar puncture to look for low CSF glucose. And I know it's a lot easier on the patient to get a blood test and look for the gene mutation, but always remember that we can always get a lumbar puncture, and really within that day, we can determine whether or not somebody has low CSF glucose. So as we were talking about the different types of mutations that we have and whether or not there's anything unique about Glut1 as far as genetics. And the missense mutations are not the only mutations that we have. We can have pieces in the gene that are missing, pieces that are inserted that shouldn't be there, and the DNA and the whole system of making proteins really can't handle extra or missing pieces of DNA. So the gene does not produce any functional protein from that copy. We can have whole gene deletions where the whole Glut1 gene is missing, just deleted. And sometimes we've even noticed we have a small group of patients that not only have the Glut1 gene missing, but genes in the area to the right or the left of the Glut1 gene. And what that can cause is not only Glut1, but additional clinical symptoms for those genes that are also missing. So we have to kind of remember that. Testing for Glut1, I know people kind of go round and round in any disorder. Anytime you walk into the doctor's office, there's lots of genetic testing, or hopefully there should be. And people are finding out that they have Glut1 by various types of tests. So two of the most common is actually just a seizure panel. So Glut1, or the SLC2A1 gene, was added to the seizure panel. There's a lot of seizure panels out there. A seizure panel is just a group of genes that somebody has sat down, looked through the literature, and found out which genes are really causing seizures. So if you're a patient with Glut1 and if you're having seizures, the doctor may have ordered a seizure panel. And usually on those panels, they're sequencing panels. So again, looking for missense mutations because they're the most common. And missense mutations and sequencing is actually the cheapest for a lab to run. They can run all panels all day long, right and left, and not spend a fortune, even though they may charge you a lot of money for it. There are also other types of ways that we can find out if there's a problem with this SLC2A1 gene. Some people have found out that they're missing the gene, and maybe the gene plus some areas on the right or left. by what's called a microarray. A lot of times, especially geneticists will just send out a microarray. And what a microarray is the lab just looks for copy number all across the human genome. It's not specific to the Glut1 gene, but copy number just means that since we get genes from mom and dad, we're supposed to have 2 copies of most everything, with the exception of the X and the Y chromosome. So are there 2 copies there? It's just looking at the whole genome, and some people have been picked up that way because of a microarray. Now there are more people running whole exome sequencing, and actually, you know, it used to be kind of a last tier test where we just tried everything else and we're not sure what the patient has. And now a lot of people are just sending out whole exome sequencing. And what whole exome sequencing is it's a sequencing, just like the missense mutations that I talked about, looking at the individual nucleotides, the A, T, G, and C, and reading the whole human genome to see if and there's 22,000 genes in the genome, to see if there's any changes in the genome that might indicate why the patient is having the clinical symptoms that they're having. So one sort of caveat with whole exome sequencing is that they sequence the coding portion of a gene. So every gene consists of exons, E-X-O-N, and those are the ones that, like I said, they're recipe that make up the protein. But there's also pieces in the gene that are basically cut out and kind of thrown out. Those are called introns. So when the technology was set up for whole exome sequencing, it was designed to look at the exons, the coding portion of the gene. So we've since realized, and this technology has been out for about 10 years, that there can be valuable information in the introns. But whole exome sequencing will not even check for that. It's not designed to do that. So it can actually, for a number of reasons, miss the fact that somebody might have Glut1. So if you think, and your doctor really thinks you have Glut1 deficiency syndrome, you've run whole exome sequencing, there's a few things that you can do. One of them is call up, you don't have to do it, but have the genetic counselor or the neurologist or somebody there at the clinic call up the lab and say, gee, I really wonder if there's a mutation in the SLC2A1 gene. Because sometimes people at the lab miss these mutations. They generally, when they look at data from whole exome sequencing, they find hundreds of mutations and they have to sort of filter through these. And if you happen to be somebody that has a mutation in that gene and it was filtered out, then you'll never know it by the results. So again, you can call the lab, somebody can call the lab and ask them to look at the SLC2A1 gene. And usually in whole exome sequencing, they also obtain blood on mom and dad. And the reason is not because we want to know what are all the mutations that mom and dad have, but we want to look at the patient's data. So let's say we have a patient that comes in, we think they have Glut1, we run whole exome sequencing and find out there is a mutation in the SLC2A1 gene, and we can, through whole exome sequencing, we can look at mom and dad's data to say, gee, well, dad's a carrier, he has absolutely no symptoms, and I'm not really sure that this mutation is causing the problem. Or they can look at the data and the data says, well, mom or dad don't carry this mutation, so maybe we need to look at this mutation a little bit further. So we use mom and dad's blood sample to help evaluate the blood sample or the data of the patient or the proband. So it's not, again, it's not that we're looking to find out all the mutations that mom and dad have. They're really just useful for the proband. It's important, so usually genetic labs and genetic testing are very expensive. Whole exome, if you pay for it outright, you can spend $6,000 to $7,000 to $8,000, and that's actually true for some of the tests that some of these labs are offering, which is kind of shameful because it's honestly not that expensive for many of the tests that the labs do, but this is what they're charging. By and large, most labs will take most of the commercial insurance, the main commercial insurance through your job. Sometimes they take managed care, which is like the Medicaid managed care. By and large, they don't take Medicaid. But just remember, and we'll try to keep Glenna and the G1D Foundation aware of which free tests are out there, because a lot of times when there's clinical trials or somebody's looking to do a clinical trial for seizures or for whatever, even Glut1, there can be free testing out there. So we want everybody to take advantage of this. If your insurance is not covering some of these tests, try to take advantage of some of the free testing out there, because it truly is free. But a lot of times, like I said, they'll use those in hopes that they can find patients for clinical trials. So you may receive a call from somebody saying, oh, there's this clinical trial, do you want to get involved? But there again, it is a way of getting free genetic testing. So one of the things that we've looked at for years is whether or not some of these mutations, I mentioned there's lots of different types of mutations, the Missense mutation, the complete gene missing, and pieces missing and pieces rearranged, but do those necessarily correlate to your clinical phenotype or the symptoms that you have? And yeah, there are some rough sort of correlations. Obviously, if you're missing the whole Glut1 gene, or you have a mutation that causes the Glut1 gene not to be produced, and many of those mutations are extra pieces in the gene, pieces missing, things that alter kind of their reading frame. Remember when we talked about how the nucleotides are all set up like the letters in a book? Well, if you're missing some of the letters, just think about it. You're reading a paragraph and all of a sudden things are missing, then you really don't know what they're saying. So those things completely inactivate or stop the production of functional Glut1 protein on that copy. You remember we have 2 copies of the SLC2A1 gene. So by and large, those things that cause the reading frame to be disrupted are going to, on average, be more affected than missense mutations, which may still create a semi-functional protein. But again, it depends on where is the missense mutation, what is it doing, what kind of amino acid change is it. So I can't say definitely that all missense are mild, and we do know that when you are missing the whole copy of the gene, that you're going to be more severe. And especially you're going to be more severe if you have the contiguous gene deletion, which is the Glut1 gene plus genes in up and down, the right and left of that. So, I know that there's many medical quality and medical availability in the nation varies tremendously from state to state, from area to area. I mean, we have some areas where people have to drive hours to get to a neurologist. There may be no genetics counselors, there may be no geneticist in the area, and I know that that's a problem. Do you have to see a genetic counselor? Do you have to see a geneticist? No. I mean, the neurologist can send out the genetics tests if many of them know what they're looking for. They may not know of all the panels that are available, but many of them are very good, are very aware of genetics, so I don't want anybody to be concerned if you can't see a genetic counselor. But certainly it's useful to see one, because we spend have a little more time with patients than some of the other healthcare professionals might have, so we can explain more of these sort of ins and outs of genetics and Glut1. So let's transition a little bit and sort of talk about what are some of the unanswered questions in Glut1 regarding genetics. Obviously, the big issue with Glut1 patients has been treatment with the ketogenic diet, and whether or not many of them find it not very palatable, and many of them to the point where they really are just not willing to take the diet, and they will suffer the seizures or whatever issues they're having from it. And obviously this isn't the best form of treatment for any individual with Glut1, but it is happening. So we are really entering an age of the possibilities for genetic treatments for all sorts of different disorders. And there are a few disorders where individuals are getting genetic treatments whereby they receive the whole functional gene. We're not at that point at Glut1 right now, but it certainly is a possibility in the future. How this works is that the gene, which we know is functioning, you have the 2 genes in the patient, one that doesn't function, one that does, And we really need in every individual more than just one functioning copy of the gene. So if we can transfer a gene to an individual, get it to function in the body, maybe it'll make more Glut1 protein. And the way this has been done in the laboratory and is being done in some disorders is oftentimes through a virus. Now, we all catch viruses, we all get sick from viruses, but this type of virus that they're utilizing is sort of stripped down from a bunch of the genetic components that it already has. So its really only property is to hold the gene, the SLC2A1 gene, gets inserted into the virus. It holds it within its genetic information and transitions it or transfers it to our cells. It doesn't incorporate into the human gene line, which is really important because we don't want it to incorporate into the germ line. Otherwise, it can cause havoc with other genes. But it just goes into the nucleus and sits in there in the nucleus and makes the protein. So that probably will, most likely will happen at some point in the not too distant future. There are all kinds of other options that people are coming out or thinking about as far as genetic treatments. What can we do to prompt the one functioning Glut1 gene to make more protein? Every gene is turned off and it's turned on and it's upregulated and downregulated. Can we do anything regarding genetics to upregulate the Glut1 gene to make it work a little bit harder? And there's other sort of possibilities. There's a lot of research being done right now in other disorders that are also applicable to the field of Glut1 deficiency syndrome. So it's not like anybody has to totally recreate the wheel because it's happening now with lots of research going on. Some of the other kind of unanswered questions within the field is just newborn screening. There are many disorders that when a baby is born, the blood is sent to the local state lab and we get a whole screen or a panel of different disorders to see if that individual is going to have a problem or going to have certain disorders. So we hope at some point in the future to also find something that we can use that would indicate that a baby has Glut1 deficiency syndrome so that they can be treated right away, because we believe that very early onset treatment is much better than trying to treat somebody when they're 19 or 20 or 10, which I'm sure that many of you have found out, especially people who have found out that their children late in life had Glut1. One other sort of unanswered question is we are aware of patients who look completely like they have Glut1 deficiency syndrome. They have low CSF glucose, they have seizures, a cognitive delay, the whole 9 yards in the most severe case. But we've not been able to find a mutation in them despite looking in many different ways at the genome. So we do know some of those people are out there. It's possible that just with all of our technology, we haven't been able to look at every single thing in that SLC2A1 gene, or it's possible that there's some other type of disorder that sort of mimics Glut1, or it's possible that it's something regarding memory. Remember I talked to you about up-regulating and down-regulating a gene. So there's lots of sort of avenues that we might think of as far as what might be happening with that individual, but we do know some of those people are still out there. So let's just change the topic a little bit and talk about, from a genetic perspective, what else do you need to know regarding your families and genetics? And I think a really important thing that I know is on everybody's mind is just family planning. What do? We want to have another child, or our child with Glut1 has become an adult and they want to have kids themselves. So I think it's really important as a family and as individuals to think about family planning so that we hopefully can have very healthy children or grandchildren, might that be the case. So one important thing is just to be very proactive about birth control. So if we know our Glut1 children are getting to be their teenage years, it is important to have the discussions about birth control because we want to be able to plan our families. So if we think about, and this is not unique to Glut1, but any genetic disorder, it's really useful to try to, again, have healthy kids. So we want to plan the families that we have rather than just not planning and then all of a sudden the 50% chance for each child of having Glut1. What some individuals choose when they know they have a genetic disorder of any kind, some people choose not to have children, some people choose to adopt, and some people choose the technologies that we have available. One of them for males, so let's just say we have an adult male with Glut1 who wants to have children. Now, what they can do is use sperm donation. So if they obviously the child will not look like them, but them as a couple can use sperm donation if the male is the one with the mutation. If the female partner is the one with mutation, they of course can also use egg donation. That is an oocyte. You go to an IVF lab and pick out the eggs that you want. There is also something called pre-implantation genetic diagnosis. And in that situation, we take the male's, again, the male's sperm, the female's egg, in the laboratory, fertilize some eggs, and do genetic testing on the fertilized eggs. And then you would pick out the fertilized eggs that do not carry the mutation and implant them. Thereby you are bypassing all sort of the risk for transmitting that mutation down. It is very important if you use some of these IVF or go through the IVF clinic or use a donor egg, donor sperm, that you get standard carrier screening, because the standard carrier screening just means that within our population there are disorders or people carry mutations in certain disorders at quite a high frequency. So we don't want to miss the fact that, so we're trying to eliminate the transmission of the Glut1 mutation, but we don't want to pick up something else. So let's be really clear that if you are using something through an IVF clinic, to have the donor sperm donor or the egg donor get standard carrier screening, so we don't have other common disorders like cystic fibrosis or spinal muscular atrophy, Fragile X. All those are common carrier screening, and we don't want, and those are also mutations that people carry in pretty high frequency in our population. So get that testing done. One other option when we think of family planning also is we can also be the individual that is pregnant that maybe hasn't used any of these techniques. We can also test the pregnancy to see where the fetus is at. Does the fetus carry the mutation? Does the fetus not? And then parents have to make a decision about what they want to do. So those are really just some options regarding family planning. All this is available. All this is, most states have IVF clinics. The only major issue is the expense of it all, because honestly, you need pretty good insurance to be able to pay for some of this, to utilize for some of these items, and you may also need to spend some money too. But just realize that there are lots of options when we think of family planning. It's not just one option for individuals, but we do want to at least send any individual who has Glut1, speak to the genetic counselor, speak to the geneticist, speak to the neurologist, and let them know that you are interested in this if you're in fact wanting to have children. And then of course just be very proactive about birth control so that you are able to plan your families. So I just want to thank everybody for listening. I know genetics is a little bit complicated, but hopefully everybody learned enough to ask the right questions to your neurologist and your doctors. You can find genetic counselors if you need through the nsgc.org website. You can look up a regular location and try to find somebody. So if you feel that you want to get to a genetic counselor, you should be able to find someone, hopefully somewhere in your area. And good luck to everybody in getting your genetics evaluated and trying to find treatments.